Welcome to Bioanalytics Lab. On this channel, I teach and demonstrate hands-on data analysis and visualization using real biological and omics datasets. You will learn practical workflows in metabolomics, proteomics, bioinformatics, and statistical analysis that you can apply directly to your own research. Welcome back to my channel. In this video, I am going to show you how to perform gene enrichment analysis in R using proteomics or transcriptomics data. I will walk you through the example data format first and then go step by step through the R code and finally explain how to interpret the output result. By end of this video, you will a clear understanding of how to run GSEA in R and how to apply it to your own omics data. The dataset contains sample information along with genes and their corresponding expression value. It also includes a condition column which defines the sample group used for differential analysis. After performing differential analysis and generating the statistical result, such as fold change, FDR, and p-values, we will then use those results to carry out Gene Set Enrichment Analysis or GSEA. This workflow applies to both proteomics and transcriptomics dataset that reflects a typical real-world analysis pipeline. This is the all required packages and corresponding libraries we need to load. So as we actually showed the data, example data, just load the data and view the result file. So this is the data. Sample information, gene information, and condition as a group. And separate the data from the group information. And first we need to do the log to transform, since actually this is the raw data. And then we calculate the log to fold change, then linear fold change. And then we will perform the p-value, t-test, and FDR calculation. And finally, we save the data in Excel and CSV. And then we perform the GSEA analysis. So this report file we got from this differential analysis. And first load this differential data. And you see this data contain p-value, FDR, log to fold change, linear fold change. So the data, and we are going to use this FDR less than 0.25. If you want to use this p-value instead of the FDR, you have to use this p-value. So we are preparing this data before going to the Gene Set Enrichment Analysis. And now we will do the geopathway analysis. And you see this, how actually the fold change and genes, corresponding genes. And first we will do the geopathway analysis, and perform the geopathway analysis. And it will take some time to finish the analysis. And once finish the analysis, we are going to save the result. save the result in our directory, and we are going to create a top 10 pathway as a dot plot. And you see this the right side in the GSEA to go geo dot plot, and save it in our directory. And we are going to perform the GSEA enrichment plot. And we are going to extract the top 2 based on the p-value. And I will show you 2 later. And then do the hallmark pathway analysis. So just to use this code as it is. Once actually you prepare your file, you don't need to do anything. And save this analysis. It will take time to finish the GSEA analysis. Once it will be finished, you can save it, and it will save in our directory folder. Save it. And again, actually, we are going to show the GSEA plot top 10. There are only 2 pathway pool. That's actually only 2 pathway. So save it in directory. And then again we are going to perform the Gene Set Enrichment Analysis. Run this code for the GSEA enrichment plot and extract to top 2 pathway. And then we are going to perform in the reactome analysis. So once you prepare your table on time, you don't need to do anything. Just run the code one by one, and then it will be saved in your directory folder, everything. And run the reactome GSEA analysis. You have to wait a little bit because it will pull all information from the library, and then you have to save it, the result. Yeah, save the result, and again show the dot plot of the top 10 pathways. Right, see the left side in the top 10 reactome pathway. Visualize in dot plot, and then perform the GSEA enrichment plot analysis. And save this plot. And here I saved as a 2 plot, but if you want to do more than 2, just change the number to 2, 3, 4, 5, 10, whatever you want. And cake pathway analysis. It is the one of the important pathway analysis for using for all cancer, non-cancer, but mostly for the cancer people using the hallmark pathway. So we ran this cake pathway. It will take a little bit time. Here I use this human database. If you want to use the mouse database, you have to choose the mouse. And once analyze finish, just save the data in directory, and we'll show here also in the cake plot in top 10 pathway. Left side you see the cake pathway dot plot in top 10, and save again in directory. And then finally we'll perform the GSEA enrichment analysis. Here also top 2 we perform and pulled. If you want to more, you can just change the number. It will automatically will saved in your directory. And you will see this the all files saved it here. So geo enrichment analysis. So plot look like this because this is not actually the high number of genes. And this is the hallmark. One of the pathway in hallmark. And here is this one of the top 2 pathway in reactome. And as I mentioned to you, it will save actually all the result. And this is the cake also. But the result, you see the GSEA, Geo, hallmark, reactome, cake. So as example, you see the cake pathway, how actually the description of this pathway, all parameter here, as well as the p-value, FDR, and how many gene materials and new content. Thank you so much.